want to thank you, the organizers, for inviting me to participate in this symposium, and also to congratulate Eduardo for his Max Planck Partner Institute, and also to the Ministry of Science, the Ministerio de Ciencia, Técnica e Innovación Productiva for the new Polo Científico, which I hope will benefit all Argentinian scientists. I'm going to talk today about work we have been doing for the past 10 years on the role of microtúbulos in signaling devices for neuronal polarization. My laboratory has been working for several years on the mechanism underlying the development of axons and dendrites, and we have mainly focused on the cytoskeleton and microtúbulos as structural elements, but more recently we have focused on microtúbulos as signaling devices. That's the reason for the title of the talk, which is Microtubule Dynamics, Rho GTPasa Signaling, and Axon Formation. And since I may run out of time, the take-home message of my talk I will try to convince you that microtubules are signaling devices that can affect master signaling regulators such as Rho GTPases in neurons during polarization. As you probably know, neurons are an extreme example of a polarized cells. In this image, you can see two pictures of a Purkinje cell on the left. On the right side is one drawn by Ramon y Cajal with his naked eye using a molecular microscope and his hand at the beginning of the century, and it shows clearly two main features of a neuron. The dendritic cardboard, in the case of the Purkinje cell, is a dramatic, elaborate dendritic cardboard, and in addition to that, the neuron has a single axon, which is characteristic of many neurons. They have a single axon and a highly elaborate dendritic tree. On the right image is a computer reconstruction from a confocal image of a Purkinje cell, and you can see also the extraordinary ability of Cajal. It's clearly much nicer cell drawn by Cajal more than a century year ago than the one generated by our state-of-the-art computer technology. In any case, this serves to illustrate that neurons are polarized cells, and this polarization arises because they are able to develop a single axon, two compartments, the somatodendritic compartment and the axon. And the axon is specialized in most cells to send information, and dendrites are specialized to receive information. This is in a very general sense. Polarization is not a characteristic, is not a proper exclusive property of neurons, but is also shared by many specialized cells, like for example here the budding gist, a migrating fibroblast, an epithelial cell, and another neuron, in this case a pyramidal cell of the cerebral cortex. Polarity has been studied at the cellular and molecular level in all these cell types using basically culture systems. And in the case of neurons, neurobiologists have also used for many years cultured neurons to study the cellular molecular basis of polarization. And the main system that people have used is a system that was developed by this guy, who is Gary Banca, when he was a postdoc at Max Cohen lab many years ago in the late 70s. He developed the culture of hippocampal cells obtained from the embryonic hippocampus. These cells, he grew these cells at very low density in a defined medium. The cells grow at very low density, so what they do was practically an expression of a cell autonomous program that could lead to the differentiation of a single axon and dendrites. Carlos Dotti and myself, we did in the early 80s a postdoc training in his lab, and we characterized a series of molecular transformation or morphological transformations that these cells undergo as they develop in vitro that lead to the differentiation of an axon and dendrites. And these stages are shown here. The cells shortly after plating, the cells are round. In green, there is staining for microtubules, and in red, there is staining for filamentous actin, for phyloidin. The cells are round. There is no evident sign of polarity at this stage. Then the cells begin to extend small neurites. This is what we call now the bipolar stage. Then they go to this multipolar stage in which the cells extend several processes, which are, there's no sign, almost identical. These processes are highly dynamic. You will see a movie. They elongate or retract, And by the end of the first day in culture, one of these processes initiates a phase of rapid elongation, becoming the neuron's axon. The remaining minor process will remain static for several days until they will begin to differentiate into dendrites. And by the end of the first week in culture, the cells have well-developed dendrites and then begin to make synaptic contacts. These are the classic 5 stages of development of hippocampal cells. And this is a movie from the laboratory of Koso Kaibuchi that shows the behavior of minor processes before and during the transformation of one of these processes into the axon. And you will see these processes will compete between this, a kind of tug of war between these processes that will lead, oops, I'm sorry. So you will see this process, they elongate, retract, until one of them initiates a phase of rapid elongation, and this will become the neuron's axon. This is a movie from Kaibuchi's lab, and this is a movie from a former PhD student at my lab. This cell, this is FICE microscopy, the other one was DIC microscopy, and here again, one process is growing. This movie was taken within a whole day, and you will see that one of these neurites will develop as an axon. It will initiate elongation. There is still tug of war in the other process, but finally one axon will consolidate and the cell will reach what we call stage 3 of polarization. So we are very interested in trying to understand how a neuron can generate a single axon from an array of symmetrical, almost identical neurites, and that have been the focus of our research for the last 10 years or more. And we know from work from biochemical studies done in the early 80s that neurite outgrowth involve at least these 4 events: cytoskeletal assembly, or let's say dynamics, and we are going now to introduce that involve cytoskeletal signaling, that there should be membrane adhesion at sites of active growth. In the case of developing neuron, this is the growth cone. There should be membrane sorting. There are proteins that are membrane proteins that are going to go to the dendrite or to the axon, and this is probably involved the Golgi apparatus. And there should be protein membrane traffic, selective protein membrane traffic to either the axon or dendrites. And we are focusing today basically on cytoskeletal dynamics and signaling. And we are not going to address any of the other points, adhesion, sorting, or trafficking. So these are immunofluorescent staining of stage 2 cells just before the axon is formed. And here is a cell that has already developed an axon. And one thing that happens in a cell that has developed an axon is that a population of microtúbules becomes stable. And this was when Mitchison and Kirchner, when Mitchison and Kirchner proposed the theory of dynamic instability. One of the predictions of this theory was that dynamic instability could be a good way in which to differentiate in time and space microtubules, and that could be important for generating cellular asymmetry. And they proposed that probably microtubule stabilization will be associated with the polarizing phenomenon. And in fact, this has been shown for migrating fibroblasts. There is microtubule stabilization toward the leading edge of fibroblasts that are migrating. And in the case of neurons, there is also microtubule stabilization during axon formation. And this is shown here. This is a cell. This cell is an hippocampal cell that has developed an axon, which is stained with antibodies that recognize alpha tubulin isotypes that label dynamic microtubules, stable microtubules, and you can see that stable microtubules are restricted to the axon but are not present in the minor process. So one phenomenon that accompanies the formation of an axon is microtubule stabilization, and the other event that accompanies the transformation of a minor neurite into an axon is microtubule assembly, and this can be seen here. One question that attracted the attention of people working on polarity was trying to identify early morphological changes that would predict which minor neuron is going to become an axon, and it took a lot of time to do that, more than 10 years to discover this, but finally work done by Dotti Toti's lab and our lab discovered that one event that precedes the transformation of a minor neurite into an axon is an increase in growth cone size, which is associated with an expansion of the lamellipodial veil of the growth cone and microtubule penetration and assembly within that growth cone. And you can see here, this is the growth cone of a minor neurite. In green are the microtubles. The microtubles end at the base of the growth cone. This is a small growth cone with a lot of actin ribs that end in the central region of the growth cone. And this is a growth cone of a neurite that is undergoing transformation into an axon. There are much more microtubles. Microtubles begin to penetrate into the peripheral lamellipodial veil of the growth cone. And it is possible to follow that doing live imaging using a marker of the tip of microtubules. One can study the dynamics of that. I show the movies, and one can quantify a lot of kinetic parameters of microtubule dynamics using this approach. One of the movies is done in epifluorescence, the other is looking at microtubules that are in contact to the subcortical cytoskeleton with TIRF microscopy. At the end of the experiment, it's possible to fix the cells and then analyze the number of comets that are labeled with this plus tip, for the plus tip map. And this shows both in vivo and in fixed tissue that microtubule assembly is one event that accompanies axon formation. And people has identified proteins that can regulate microtubule assembly in these neurons, and one such protein is, for example, this protein CRMP2, which is collagen receptor mediated protein 2, which can drive microtubule assembly. Its overexpression induces the formation of multiple axons in these cells, and its suppression by RNAi inhibits axon formation. Aside, there are many other proteins that can also drive microtubule assembly, but this is a very good example of a protein that regulates axon microtubule assembly during axon formation. So please note that overexpression of this protein in this cell system can induce the formation of multiple axons. So in this model system, in the hippocampal cell culture, which only extend a single axon, one idea is that all minor processes have the potential to become an axon, and the question is, how come only one axon is formed? Another experiment important and related with that was published in Science in 1998 by Carlos Dotti and Frank Bradke, showing that it is possible to induce the formation of a single axon by depolymerizing the actin cytoskeleton with very low doses of acetylcholinesin D. And we did a similar experiment using global application of acetylcholinesin D and induced the formation of multiple axons. Now, so in addition to microtubule assembly and microtubule stabilization, there should be an increase in actin dynamics. Is another phenomenon that accompanies transformation of a minor neurite into an axon, and this is shown in here in this image of a growth cone. Now you can see that actin, which is stained in red for phalloidin, is not anymore organized into actin ribs. Most of the actin is loose, organized in a loose network, and microtubules have penetrated all toward the middle region of the growth cone and to the periphery of the growth cone. So when a minor neurite becomes an axon, it's going to become an axon. There are all these events. There is microtubule assembly, there is microtubule stabilization, there is an increase in actin dynamics, and there is microtubule penetration into the central and peripheral region of the growth cone. And interesting, there is microtubule capture at subcortical cytoskeleton. And this is a very important phenomenon because microtubule capture can be associated with deployment of factors that are bound to microtubules and can regulate the activity of other signaling regulators like Rho GTPases. All this work came to the idea that microtubules and actin filaments are coordinated, regulated during axon formation, and the question was what coordinate the actions of microtubules and microfilaments during axon formation, and the question was very simple. People read the conclusion that this may be achieved by Rho GTPases of small Rho GTPases like RAC, CC42, and Rho. As you probably all know, these are molecular switches that go from a GTP-bound active state to a GDP inactive state, and when they are in the GTP-bound state, many molecules associate with them and regulate downstream effects. And the general idea is that RAC and CEC42 are positive regulator of axon formation, and that Rho is a negative regulator of axon formation. And there are several lines of evidence, I will go very fast, that indicate that this is the case. For example, activation of the IGF-1 receptor leads to activation of CEC42, and CEC42 is required for axon formation. That we published several years ago. Now is accepted. There is threat analysis show that there is a strong CEC42 signal in the axonal growth cone at the time of polarization and during axonal elongation. Similarly, a similar situation is in the case of RAC. Indeed, overexpression of RAC leads to multiple axon formation, and suppression of RAC inhibits axon formation. Rho has the opposite effect. Inhibition of Rho A activity leads to multiple axon formation, while overexpression of Rho inhibits axon formation. And during polarization, threat analysis show that there is low Rho activity in the future axon and high RAC activity in the future axon. And the question arises how these activities are regulated, and candidate factors for this are a group of proteins now that regulate Rho GTPase activity, which are guanosine nucleotide exchange factors. And since I think I'm running out of time, I will just summarize part of the talk just by saying that there is a balance between Rho and RAC CEC4 activity. Rho is a negative regulator of axon formation, while RAC and CEC42 are positive regulators of axon formation, and that microtubule-associated HEFs, one in nucleotide chain factor, regulate the activity of both Rho A and RAC and CEC42. An example of a microtubule-associated factor that regulate Rho activity is LFC, which is a HEF for Rho A. LFC, also known as HEF-H1, is very interesting because this protein becomes activated upon microtubule depolymerization. So microtubule depolymerization activates LFC. LFC activates Rho A, which by means of Rho kinase inhibits the RAC CEC42 part 3, part 6 complex, inhibiting axon formation. So some way the cells need to get rid of the activity of the LFC Rho A activity, and this is achieved by one protein which is also associated with the microtubules, which is a dynein light change. I will pass several slides.